So quiz today. I forgot to bring that up on Tuesday. Quiz opens today. Standard. Open 5:30 and then run till tomorrow 10ish. Big one, 6 questions. So take your energy drinks beforehand. Questions, concerns, let me know. Anything before diving into today's lecture? Nope. All right, so kind of part 2, if you will. Yeah, it's bugging me. So, I should have done this before getting up. That was horrible. So, furthering discussion on cocaine and stimulants, but in terms of polydrug use, because until recently, really, cocaine was the most co-abused drug. Well, cocaine and alcohol were the most co-abused drugs around. That landscape is definitely shifting into the new designer drugs. But as this mentioned, polydrug use is just using multiple substances at the same time. Very common recipe, if you will, are mixing depressants and stimulants. As I mentioned, cocaine and alcohol was the biggest one. Nicotine in everything was used for the longest time, but now isn't so much. Mixing of a depressant and a stimulant, especially cocaine and then an opiate, especially heroin, was the big one. Known as speedballing, was a really big party drug for a while. But that was more for the heavies, I guess. Cocaine and alcohol were the very standard kind of socially mixed drugs for a number of decades. But again, both of those have just fallen out of favor recently. So this is another lecture. It's going to be very different 5 years from today than it is now. But the idea again is that mixing multiple drugs at the same time is a very common practice in the party world of today, if you will. But we'll talk about not just mixing them together, but how they can potentiate each other, or the actual physiologic response or pharmacological responses that can happen when using more than one drug together. So that's where we're really going. But as I mentioned over and over, cocaine and alcohol were the most commonly co-abused. And the reason for it really was because they went hand in hand for the most part. And the idea was that if you drink alcohol, you start to have a few drinks, you start to feel good, but then after a while, you start to taper off. And you start to really lose your energy, you start to have a dip in your energy, and then applying more alcohol doesn't really bring you back up. But what does will be cocaine. So cocaine can pick you back up and keep you partying well into the night or days of binging, whatever level you're at. On the other side, say you start with cocaine and you take some cocaine and then you get up, and then you take some more cocaine and you get up, but then the third time you take it, you're not as up, but you're starting to get really jittery, but you're still looking to get up, but then you want to take more cocaine, but you're getting even more jittery and you don't like it, so you need something to help you out, so you figure you have a drink because it'll calm you down. So then you have a drink with that cocaine, and then you have more drinks. So it's really the basis of how these two drugs affect each other really contributed to why they are co-abused. Because one will bring you up and one will bring you down. But one that brings you, you can feel up by alcohol, but then eventually it'll make you feel down and you can't really drink your way out of that. You need something that really picks you up. The other side is also true. You get up from cocaine, but then repeated doses of it will make you feel less euphoric and more just jittery. So you're looking for something to calm you down, and that's where alcohol comes in. So, big reason why these are co-abused, and unfortunately treating these, once a person gets used to that sort of cycle where you become dependent on both of them, really makes things hard to treat. Yeah, for various reasons. So, one thing that's very interesting about cocaine, though, is binging with this drug, all right? So, I don't know if saying cocaine is less lethal than opiates, but has potential to be less lethal. However, when lab animals are given an unlimited supply of of cocaine, even compared to opiates or just heroin, lab animals will tend to binge on cocaine until they die, but they won't do it with opiates. Now, there is certain toxicity that we talked about with cocaine and stimulants. We'll go over it again in this lecture. But the toxicity of opiates is definitely more profound because we see it a lot more often, all right? And however, what is very interesting in this field is that even though cocaine tends to be less lethal in terms of how the drug physiologically affects you, binging on the drug is more prominent in cocaine than in heroin. And especially if you're given a kind of a wide open space to binge as much as you want, laboratory animals have shown that they will binge on cocaine until they kill themselves, and they won't so much with heroin. As shown here, when looking at, so again, hypothetical data as we usually look at, number of injections per hour, and then given unlimited availability to cocaine, what we see is days of unlimited cocaine access. Animals will press bars, to get an injection of cocaine, and they'll do it till they don't feel anything anymore, right? They've kind of saturated, they've either hit a synaptic fatigue, if you will, or a point that taking more cocaine doesn't do anything to them. So they drop off and they stop pressing the bar. But then they start to feel, the synaptic vesicles fill back up, or they get more neurotransmitter that's ready to fire, so then they go back up, they press it again, and they repeat this cycle until they get to that place of death, till they kill themselves on this drug, all right? Comparing that to the same situation of an animal with heroin, we see the animals can get to that state with heroin, but it's much more likely that they do that with cocaine. This is still not entirely understood phenomena, but it's something that you can demonstrate very readily in a laboratory. So the idea of binging is a much bigger issue with cocaine than it is with heroin. Even though you can have respiratory depression and death with heroin, much more common than with cocaine, it seems like the animals get to a sated state of euphoria with opiates where they just kind of either taper off and pass out, or they just don't look to have the reinforcement that they do with cocaine, to a place where they, it becomes very toxic. So anyways, interesting aspect of cocaine that kind of separates from others. Cocaine accounts for about a quarter of the non-fatal myocardial infarctions in young adults. And this is really, these numbers are changing for sure, especially due to the fact that the modern landscape is really all about opioids, and designer drugs are steered more towards that type of class of drugs. Stimulants aren't so much in vogue as they used to be. So again, the toxicity with this really comes from the norepinephrine tone in specific areas of the brain, as we discussed before. So common figure in this class, presynaptic, postsynaptic nerve terminal. Here's our synapse. We're discussing norepinephrine here. And when talking about adrenergic receptors specifically, the body has these in sovereign spaces, so you don't have, you rarely have overlap in terms of stimulating the same sets of receptors that are doing conflicting things at the same time, all right? The body keeps that very tightly regulated. However, if we dump a whole bunch of a stimulant on top of it, it doesn't have that ability to keep these things sovereign. We're just gonna release norepinephrine wherever. you have these sorts of substances present. And as I discussed, when stimulating beta-style adrenergic receptors, you can increase the heart rate. Stimulating alpha-style adrenergic receptors can cause vasoconstriction, and these are two things that you don't want to happen at the same time. You're increasing the flow or the flow of blood through the circulatory system, but you're constricting the blood system too. The overall effect of that is going to be a massive increase in pressure on the system to an extent that something is going to have to give, whether it's the sovereignty of the vessels or the heart itself. So this is where big toxicity comes into play. And this just sort of, it's kind of a confusing situation because you can have, depending on the area of the body and depending on the specific type of receptor, you can have vasoconstriction or vasodilation. But as this shows here, the alpha receptors will be responsible in certain areas of the body for providing vasoconstriction. Beta receptors will provide vasodilation. That makes sense, hopefully, as you think about previous slide. If you're going to increase your heart rate through beta stimulation, you would want to dilate the blood vessels, right? You'd want to give larger area for more blood to go through. If you need to oxygenate your tissues, you want to have more rapid heartbeat, more beats per minute, if you will, but you'd want vasodilation there, so it doesn't cause that increase of pressure that can blow back on the heart or on other vessels, all right? Okay. Why this is also important is not just for the toxicity aspect, but we're going to add another layer to our control of the reward pathway, okay? So really throughout this semester, we've been constantly building on this figure right here, right? We've got our dopamine neuron that's central to this whole thing. Soma is in the VTA, projects out into the nucleus accumbens as well as the PFC. We'll synapse on dopamine neurons here, glutamate neuron in the PFC. Once dopamine is released, it'll signal through D1. The glutamate here can feed back and through activity at AMPA-A receptor can turn on dopamine to make a full circuit. We talked about this being under the control of a GABA interneuron here. That's when the GABA is on, dopamine is off, but when GABA is off, dopamine is on. Adding another realistic layer of control here, we see this is the same, here's the same thing here. So here's in the VTA, this is our dopamine neuron. So this neuron right now is this neuron right now. Synapsing on nucleus accumbens and then PFC, that's feeding back with the glutamate, right? But now we're seeing the locus coeruleus input here. This is feeding norepinephrine onto this dopamine neuron. So right here, we would have incoming norepinephrine with an alpha-1 receptor on dopamine. So it's going to be under the control of two different impulses, right? Or two different inputs, excuse me. Then also we have a projection off into the PFC here. So this norepinephrine can also be controlling the glutamate input here, okay? How does that look, everyone? Okay, so all we're really doing here, we've got our standard schematic of our reward pathway. We're just adding one more layer of control where norepinephrine projections from the locus coeruleus can turn on these neurons as well, right? And this is important because norepinephrine signaling has been shown to contribute to cocaine reward. So just as we described that pathway there, you can show that in experimentation. As shown here, we're plotting time in a chamber. So we're looking at a condition place preference, only part of it here. So again, this is the one where you can steer an animal into a desirable or less desirable side of the chamber based off of where you're feeding cocaine, okay? So wherever an animal is getting cocaine, that's the side it's going to want to spend more time on just due to the fact that it's getting cocaine there. So looking at a wild type mouse with no genetic alterations, where it's getting cocaine in chamber 2. In chamber 1, it's just getting saline, so it's going to want to be in chamber 2, right? However, if we knock out dopamine beta hydroxylase, the enzyme responsible for converting dopamine into norepinephrine, we see this effect is blunted, all right? Knock out D beta H, you don't have a preference for either side, meaning these mice or these animals are not getting the reward pathway turn on and that reinforcement that they want to be on that side where they're getting the cocaine. So they don't have a preference of which side of this chamber they're on, because whether they're getting saline or cocaine, they don't feel the difference, all right? So norepinephrine signaling is important for developing cocaine reward. So to bring in the idea of polydrug use, we can also talk about nicotine use, as this can potentiate people for developing behaviors that lead them towards cocaine use, dependence, and addiction. As this shows here, we're looking at percent of the population who are cigarette smokers and caffeine drinkers, whether they're just general population or cocaine dependent, right? So the filled in bars are going to be cocaine dependent. The open bars are general population. And what we can see with caffeine drinkers, there's really no distinction between whether they use cocaine or they don't. People who use cocaine, some drink caffeine, some don't, doesn't really matter. It's kind of an equal distribution. But where it's really, where you see a very close tie between polydrug use is in nicotine abuse or cigarette smokers. As this shows, a vast majority, what we're like 4 to 1-ish, people who smoke cigarettes, I should say that differently, people who are dependent on cocaine are much more likely to smoke cigarettes than to abstain from cigarettes, okay? So this just shows a correlation between nicotine use and cocaine use. But what's more important, and I thought I took this out, sorry. So this is essentially another study showing exactly what we just showed on the previous slide, comparing smokers and non-smokers to those who use cocaine. The biggest thing here is you'll use a lot more cocaine if you're a smoker than a non-smoker, but this just re-emphasizes the correlation between cigarette use and using cocaine. This is what I wanted to get to. Okay, and we can really see some effect when we're talking about age distinctions between younger and older animals and their development of cocaine use and abuse, and how that can be altered with nicotine, okay? So we are looking at, we're doing observation chamber here, and we're looking at how much a mouse walks around in a chamber with different types of treatments and pre-treatments on board, okay? So ambulatory counts, this is just the number of steps that a mouse takes in the 30-minute session, from 0 to 800 steps, okay? And so what we're looking at, this top group, what A and B panels, are adolescent mice, C and D panels are adult mice. We're gonna pre-treat these animals with either saline or nicotine. And then while they're in the observation chamber, they're gonna get boluses of either cocaine or saline, okay? So just looking at the left saline pre-treatment, we're just discussing the differences between how cocaine will affect an adolescent versus an adult mouse, okay? So black squares are the animals that are getting cocaine, white circles are animals that are getting saline, and we can see that there looks like giving cocaine to an animal, an adult animal, is more impacting in terms of how much they walk around compared to an adolescent. because we can see here, it looks like the saline treatment is having somewhat of a stimulatory effect here, right? Especially compared to adult. All right. So, control treatments with saline, we have more walking around when we give cocaine, whether if it's an adult or an adolescent, right? Seems to have more of an effect on the adults, but the bigger picture is that we're seeing more walking around with cocaine than we do with saline. We saw that last time. This is not too profound to state. However, if we go in with nicotine, we pre-treat these animals with nicotine, as you can see, there's a much more profound effect on adolescent mice compared to adult mice, all right? And this is the bottom line here. So pre-treatment with nicotine predisposes adolescent animals towards cocaine dependence and abuse more than adults, all right? So the fear obviously is that, you know, nicotine is one of the most common drugs that youth get exposed to, right? Used to be through cigarettes, now it's more dipping and vaping, if you will, but yeah, they're still exposed to nicotine, and that's one. Cocaine and caffeine are the two most commonly, two drugs that youth are most commonly exposed to. But what we see here is it's not just the nicotine itself that is a concern. It's the potentiation towards cocaine abuse that we see from studies like this. So again, pre-treatment of an adolescent animal with nicotine prior to cocaine treatment causes an increase of the effect of the cocaine in terms of the locomotor sensitization on that animal, right? How does that sound? Right. So the same thing applies to the reward pathway here, right? Before we talked about how, or last time we talked about, you know, how much cocaine makes people move around versus how much it turns on the reward pathway. We're seeing the same sorts of thing with nicotine treatments in both the locomotor activity as well as reward pathway turn on. So what we're seeing here is our conditioned place preference. We're giving cocaine in chamber 2, and we see that when animals were pre-treated with nicotine as adolescents versus as adults, it has a much more profound effect with how the cocaine stimulates the reward pathway compared to adults, all right? So cocaine's turning on of the reward pathway is much more prominent in adolescents that have been treated with nicotine compared to adults treated with nicotine. This can also be tracked back to the FOS proteins that we talked about that are important for molecular responses to cocaine in any animal, right? Specifically, the delta FOSB protein levels can be altered in the same way. So again, we're talking about adolescent treatment versus adult treatment. Saline controls, you have a certain amount of delta FOSB protein levels. In adolescents that are pre-treated with nicotine, you have much higher delta FOSB levels than are adults that are treated with nicotine, again, okay? So, one of the clues as to how this happens at the molecular level is an increase of the delta FOSB protein levels that have been shown to be important for development of cocaine addiction, important for how the cocaine actually affects the system. And we can see here that this also comes, or nicotine treatment also comes into play with the same system. Okay, so moving into actual treatments for cocaine, due to the fact that cocaine and alcohol are so commonly co-abused, The idea that we could use disulfiram for treating co-abusers of alcohol and cocaine came up due to the fact that, so again, disulfiram inhibits aldehyde dehydrogenase, right? If you give it to someone before they drink alcohol, they get all kinds of sick because they're filled up with aldehydes and those are nasty chemicals. But disulfiram can also inhibit dopamine beta hydroxylase, right? The enzyme that converts dopamine into norepinephrine. So this drug was thought to be able to be a treatment for someone who is co-abusing both of these drugs due to the fact that it hits both enzymes, or two different enzymes that are involved in the response to alcohol as well as cocaine. However, biggest problem here is that disulfiram also inhibits carboxyl esterases, one of the enzymes that's responsible for breaking down cocaine. And as I mentioned last time, when you have cocaine and ethanol on board, this can be converted into cocaethylene, a highly neurotoxic and hepatotoxic compound. So, this idea never really got a whole lot of traction just due to the fact that they could see the science works out, or the theory works out, that disulfiram could help. But the problem is, if you already have someone with ethanol and cocaine on board, if you start steering their metabolism pathways into development of these highly toxic compounds, you're going to really hurt the people that you're trying to help, essentially. And yeah, they'll be more prone to emergency room visits based off of these toxin buildups than potential overdose situations. So, just a bad idea in general. But so I mentioned last time that we don't have a specific treatment for cocaine abuse. And that is true, but misleading, because I should say there are drugs that are used for managing symptoms of cocaine abuse, okay? We don't have a drug that specifically targets cocaine or tries to block or prevent the use, similar to methadone and opiate use, okay? So benzodiazepines, as well as monoamine modulation, so SSRIs, SNRIs, those are being used for treating people with cocaine addiction to a certain amount of success. I mentioned before CBD, or cognitive behavioral therapy, CBT. That's a big one, trying to distract people, but very low rates of success. People really have to want to be off of cocaine in order to get off cocaine. A number of other small molecules have been tried with very little success. So dopamine antagonists and D1 agonists, so things that would replace cocaine at the dopamine transporter, as well as things that would stimulate the D1 receptor. Biggest problem with those are this does exactly what cocaine does. So people get the same effect of the cocaine as or the same effect with that antagonist and D1 agonists, so dopamine 1 receptor agonists. They get the same effect using those as they do with cocaine because they have the same mechanism of action. So the problem is they become addicted to the treatment, and you really just trade one problem for another. Vaccinations is something, we'll talk about that for the next few slides, but the idea that you could either deliver a vaccine against cocaine to a person so that it would either sequester it in periphery or break it down in periphery, depending on the type of antibody. Or you could give someone a cocaine-like analog and let their body develop natural antibodies to that. Both of those situations have been tried, and we'll talk about the success for that. So antibodies talked about. Enzyme-based therapeutics, we'll get into that at the very end. So actual proteins that can break down cocaine in the periphery are also being explored as honest cocaine therapies for either cocaine overdose or for addiction. So in terms of immunization against cocaine, one approach, as mentioned, is to deliver a cocaine analog to an animal and then have it develop antibodies against that. that can then, upon subsequent administrations of cocaine, the thought would be that the animal would then have a nice spike in antibody levels that would fight off that the cocaine. And here's some numbers. So GNC is a cocaine analog, KLH, keyhole limpet, hemocyanin is an adjuvant, if you will. So this just kind of spikes the immune system or primes the immune system to go into having B cells produce antibodies. So we're looking at the cocaine analog used to immunize rats versus control, which would probably just be KLH with nothing. So the numbers here that are important. So you see number of cocaine bar presses here. So baseline compared to extinction. And then after they've been immunized and have had exposure to cocaine. So the bottom line here is that the higher the number, the more they're going after the cocaine, the more they're feeling the cocaine. Okay, so in the control, you can see they have roughly 15 different bar presses in their observation time. And here it's only 4, less than 5. And we can see in both days, we have much less desire to bar press to get a cocaine injection or administration. So the idea here is that this immunization allowed for antibodies to be produced that would then take away the cocaine in periphery so the animal wouldn't feel the effects of it. They showed that it worked here, but unfortunately it's not a very good approach, as what they found long term is that you can show in very selective studies like this that an antibody against cocaine will decrease the amount or the response to cocaine. But what they found in humans is that when you know all you have to do is outcompete some antibody, you'll buy more and more and more cocaine. You'll take more and more and more cocaine until you outcompete, or you have more cocaine in your system than you have antibodies that'll sequester it. So unfortunately, this approach really fell by the wayside. Similar here, but the opposite approach. So GNC92H2 is an actual antibody that was developed against cocaine that was then shot into circulation in these patients we're looking at here. Well, looking at a 1-hour session, different doses of the antibody against cocaine compared to just control IgG, we can see the white bars, there's the same number of infusions per 1-hour session because we're not seeing any effect whether we have a low or a high dose of this control IgG antibody didn't have any effect. Higher doses of the anti-cocaine antibody show that you'll have less infusions per hour, meaning that this antibody is present in large enough quantities that it's able to sequester the cocaine in periphery and you don't feel the effect. So whoever's doing this infusion isn't feeling the reward pathway turn on due to the cocaine infusion, so they just don't press the bar, don't go for it here. But, so higher levels of this anti-cocaine antibody show that it can curb the behavior. Bottom line though is the same thing with the other type of immunization, where if you just take more cocaine, you completely obliterate this effect. So similar situation, looking at vaccine in humans. 115 crack cocaine users were randomized into vaccine or placebo groups. There was vaccination, placebo or succinyl norcocaine, which was used to develop the antibody against. And what they found out is having a titer or a level of antibody greater than 43 micrograms per mil would allow for these patients to have cocaine-free urine samples, okay? So this is another study that established an actual level of antibody that you would need in circulation in order to curb people from using cocaine. And they set that as kind of the gold standard, that you had to have above 43 micrograms per mil of circulating anti-cocaine antibody in your system in order to work, but the bottom line, oh man, okay, I'll go back to that. What they saw then is when they actually tried to open this up to a larger amount of people, and they went by their greater than 42, 43 micrograms per mil of antibody titers, it had no effect on the long-term validation of this study. So what we see here is that whether the people are the placebo control or the vaccinated cohort, they all come in with a certain positive amount of urine after rechecking after the study, just due to the fact, again, that people, once they know that they have an antibody in circulation and they can outcompete it by just doing more cocaine, that's exactly what they do. I apologize for skipping over this. This was the setup for a study, and I put it in here really as more of an aside. If you're interested in how clinical studies actually get set up and kind of break down, this is a very realistic view of this, where they had 736 subjects initially, and they immediately booted 426. Some of them just didn't have cocaine, and that was really confusing. I don't know anyone who would lie about using cocaine to be in a study. I mean, there must be some monetary gain that you could get. A lot of them were just not motivated to stop cocaine use, and some of them health were so bad that they couldn't be part of the trial. But this is a very common thing. You start gathering your subjects, you get a cohort of 736, and you actually start with 310, right? And then you split those into roughly 150 each, and then give different breakdowns and blah, blah. You can go through the rest there. But this is kind of a realistic setup of how an actual clinical trial would be set up. And unfortunately, a lot of the limitations to it, because that's so common that you say, you know, you gather this huge cohort, you're going to start this trial, then when you actually start to dig through and exclude people for fitness, punting half of your volunteers is very common, unfortunately. So, okay. All right, last thing we'll talk about is the biologics-based or the proteinaceous more based types of potential treatments for cocaine abuse. So this is a structure of a protein called cocaine esterase, all right? This protein was actually isolated from a rhodococcus bacteria that grows in the soil around the roots of the coca plant, all right? And interestingly enough, they believe that the plant or the bacteria developed this enzyme because it can actually break down cocaine in the same manner that butyrylcholinesterase does into this ecgonine methyl ester, the EME, and the benzoic acid. So these are very inert compounds in our body that we can just shed, but these compounds can be used as carbon and nitrogen sources for something like a bacteria. So that was the thought, is that this bacteria gets the cocaine from the soil that gets expelled by the roots of the coca plant. It can take the cocaine up. This enzyme will break it down into two digestible molecules, and it uses that as a carbon and nitrogen source for its metabolism and catabolism. But the idea here is that this enzyme actually breaks down cocaine so rapidly, much faster than any of our endogenous enzymes. If you have a person who is overdosing on cocaine, you can administer this to them through an IV shot, just like you would do like an adrenaline shot. Give an IM injection of cocaine, and the people will stabilize like that. People go from the clonic phase where they're seizuring and they're about to have their final infarction, and then this protein works so fast that it chews up cocaine in circulation, pulls it down through the blood-brain barrier, and then chews it up. And you'll, honestly see animals that are seizuring and ready to die, and 5 minutes later they're walking around the cage as if nothing had happened to them. So it's an excellent rescue tool. On the other hand, it's been engineered well enough so that if you have this protein in circulation and someone takes cocaine, it works so fast that it'll chew it up before it gets into their brain in appreciable levels. So the idea is that you can use this as a rescue tool for someone overdosing on cocaine, as well as a preventative measure. If you give this to someone, they have it in circulation, they go use cocaine, they really can't take enough cocaine to outcompete the activity of this enzyme. And yeah, so it has tremendous potential as a future therapeutic use. And the last note on this is what's really amazing about this specific protein itself is I talked about here we've got a picture from the steppes of the Andean mountains. We got a bunch of these coca plants growing here. We talked about in the leaves of the coca plants is where the cocaine is synthesized. And then the bacteria grows in the soil of these coca plants. And it's just pretty amazing that nature gives us this drug that can be very harmful to people. And right in the same exact biosphere that the drug is produced, the cure for the drug is produced. And it's kind of an amazing little aspect of nature, but whatever. So, all right. That's it for this one. Best of luck on the quiz. Let me know if you have questions. Hope everybody has a good weekend. See you next week. How do?